Okay, good morning, everyone. I'm Edward Castañeda, Florida International University, and I'm going to provide a quick overview of the herbaceous vegetation and soil data that we collected as part of the Delta X project. I'd like to acknowledge Elena Solohin, my postdoc, basically the contribution to this project, collection, processing, and managing of data sets and publications. As Robert mentioned, we have 6 sites across the Achapalaya and Terrebonne Basin. The Achapalaya is the active delta, and the Terrebonne the inactive delta. The sites that you see in blue and in red are along a salinity gradient from freshwater to brackish and saline. Our sites are located close to the CRIMS station that you see the number right there. 5 of our sites are part of the CRIMS station, and except the Waxley Delta in Mike Island, that is not a CRIMS station. As part of the inventory of data that we have assessed seasonal and spatiotemporal changes in above ground biomass and necromass or herbaceous wetlands in these sites. Also, above ground vegetation structure, below ground biomass and necromass, also soil properties, changes with soil depth as well. And also we determine foliar and root content of stable isotopes, carbon and nitrogen, that we can use that information to assess the phytoenvironmental conditions in vegetation dynamics. This is one example of the website for the above ground biomass and necromass. Basically, what you can see is in each data set, you can access the data, the methodology that we use, the sampling processing, and the CSV file, and also the citation for that specific data set. In the case of above ground biomass, basically, we have 4 stations that they were established perpendicular to the wetland edge. So we had 2 stations at 25 meters and 2 stations at 50 meters. The sampling stations were located 30 meters apart, and the idea was to capture the spatial variability in above ground biomass and necromass in the intertidal zone. Again, we also collected samples to assess total foliar carbon, total foliar nitrogen, and phosphorus. So within these permanent pots that we harvest, basically 0.25 square meters, we harvest all the vegetation above the soil, and then we sort the vegetation between life and death, and also by species composition. Here is just to give you a short overview of the actual Excel file, when you can see the columns. So we had the basin, we had the Achapalaya and Terrebonne. Column B is the campaign, so we sample in the spring and fall to 2021. We also had the lag and long location of each of the plots that we harvest, the specific site ID, and the hydrogeomorphic zone, as Robert mentioned. So we have basically the intertidal zone, except in Waxley Delta when we have a clear zonation pattern, and we have high intertidal and also intermediate intertidal zones. So sampling station, quadrats, so within each station, we have duplicate quadrats that we harvest. And then again, you have column J, you have a species. Then if it's above ground, biomass or necromass, AGB versus AGN. And then the total mass that we have per plot. And then the content of carbon, nitrogen, and phosphorus for that vegetation. Here you click in the user guide, so it's gonna give you a comprehensive overview of the methods that I mentioned already. So that's standard for every single data set. Some of the data that we are generating here, we see changes in above ground biomass and below ground biomass across all those sites. Also, you see the seasonality changes between the early growing season that we sample in March, and then during the peak biomass season that we sample in August, beginning of September. So you see the carbon allocation partitioning between above ground and below ground biomass. Also, one thing that is interesting is, for instance, the 21 site in Terrebonne Bay is the most productive site when we had the highest above ground biomass and necromass. So this is kind of the data that you can generate out of this data set. This is for vegetation structure. So basically, within each of the quadrats that we harvest, before harvesting, we assess the vegetation structure. We register the species, we measure the stem diameter, the height of the shoots, and also the stem density. And at the bottom part of the slide, you can see again some of these variables right there that we collected. For below ground biomass, we collected 50 centimeter cores, and the cores were sectioned into 10 centimeter intervals. And the idea was to assess the spatial distribution of roots. Once the roots were clean, we separated between fine roots, which is less than 2 millimeters, and coarse roots greater than 5 millimeters, and also necromas. In the right-hand side of the slide, you can see some of the samples that were already sorted out. Some of the data that you can plot, here we had changes in biomass with seasonality between the early growing season and peak biomass season, and also the distribution with depth. And what we found basically in the peak biomass, we had the highest allocation increases, but it's higher in the top 20 centimeter of soils. And that was more conspicuous in the saline size in both basins, 322 in the Atchafalaya, 421 in Terrebonne Basin. For soil properties, we have a similar methodology that LSU did. I mean, we used the Russian peat core. We collected 50 centimeter depth cores in each of the stations. We sectioned the cores every 10 centimeter, and we assess bulk density, organic matter content, and total density of carbon, nitrogen, and phosphorus content. Here is some of the data that we are generating. In the x-axis, you have bulk density in grams per cubic centimeter, and then organic matter content that we use the loss of ignition to assess the organic matter content, and then distributions with soil depth. So the pattern is that when you have low bulk density values, indicates that you have high organic matter content. And you can see really distinction in the Atchafalaya when the wax lake delta, which is the blue, you have the high bulk density relative to the fresh and the breakage. And then this is the influence of the riveting input into this system. And then this is just basically to show you the DOI for all the data sets that we have published so far. And with that, that's it.